While talking about the flow of information from the DNA to the protein, we have seen that there are two processes that are involved. First is the transcription, that is formation of RNA from the DNA, and the other is translation, that is in which the polypeptide chain is organized or laid down according to the nucleotide sequence on the RNA. So in this video, we will be talking about the first step, that is the transcription. The enzyme involved in the formation of RNA from the DNA is RNA polymerase. So it is the key enzyme, and before proceeding forward, let us understand its basic structure and the functions of these units that together form this holoenzyme, that is RNA polymerase. So in case of prokaryotes, this RNA polymerase, it is made up of 5 units, 2 alpha subunits, 1 beta subunit, 1 beta prime, 1 omega. These 5 subunits, they together form the core enzyme. And apart from that, a sigma factor is required so that these subunits, they may organize and thus form the holoenzyme. So catalytically, or the catalytic activity of this enzyme is because of these 5 subunits. Here the alpha subunit, it exists in the form of dimer, and it is responsible for the basic assembly of this RNA polymerase and its interaction with the transcription factors involved. Then this sigma factor, it basically recognizes the promoter sequence to which this RNA polymerase has to attach itself, or it recognizes the gene which is to be transcribed. This omega, it is the smallest subunit, and it is non-essential, that is, it is required, it is basically a chaperone for this RNA polymerase folding, and without it also RNA polymerase, it can perform its function, that is, it can transcribe RNA on the DNA template. Now these two subunits, that is beta and beta prime, they form a complex. They are responsible for the attachment of this enzyme on the DNA duplex. Then they are responsible for the formation of messenger RNA, and they are also responsible for the movement of this enzyme on the DNA duplex so that it may be transcribed from the 3' end to the 5' end of the DNA strand. So this entire process of transcription, it can be subdivided into 3 phases. First is the initiation, second will be the elongation, and finally the termination. So for the purpose of initiation, that is starting of this process of transcription, this RNA polymerase, it organize itself, it attaches itself to a special sequence on the DNA duplex that is known as the promoter region. It binds itself to the promoter region, and this promoter we know is recognized by the sigma factor. So once it binds to this promoter region, it basically breaks this hydrogen bond between the complementary base pairs in this region of the promoter, and this makes available the template strand, that is the 3' to 5' strand, where the ribonucleotides, they may form the complementary base pairs and initiate the process of transcription. So it gradually forms it unwinds this DNA duplex, resulting in the formation of this transcription bubble. Now this promoter site within this DNA molecule, which lies upstream of a particular gene. Now in case of prokaryotes, this promoter sequence, it consists of two consensus sequences which lie upstream the start point, that is the point from which transcription has to start itself, that is the ribonucleotide, it will form the complementary base pair from this +1 point. So upstream, that is towards its 3' end, approximately 10 nucleotides upstream, there is a Tata box. Tata box because it is rich in thymine and adenine nucleotides. It is also known as the Pribno box. And another consensus sequence in E. coli particularly, and in most of the prokaryotes, is this TTGACG box, which is approximately 35 nucleotides upstream, and these region together, it forms the promoter region. Now since this point is rich in adenine and thymine, which we know they are uh, connected to each other, joined to each other in the two complementary strands of the DNA duplex by means of double bonds. So this is the weak point, and it is easy to form the bubble, the transcription bubble, at the point that is rich in adenine and thymine nucleotides. So the sigma factor, which is produced as a result of the different signal present in the environment, it recognizes a specific promoter, that is the promoter specific for a particular gene, and it attaches itself. And this sigma factor is then recognized by the RNA polymerase, the other subunits of RNA polymerase, and this holoenzyme, that is the core enzyme, along with the sigma factor, they are uh, organized, or they bind to this DNA molecule in this particular region. Now once this transcription bubble is formed, the RNA polymerase, it attaches itself, binds itself to this DNA double helix. It will start its function, and its function, as we know, they include opening up of this DNA double helix to form the transcription bubble, moving ahead from the 3' to the 5' side, and on this template strand synthesizing ribonucleotide chain, that is a polyribonucleotide and RNA is formed. Here one more thing we should again talk of, and that is that the RNA polymerase does not require any primer. RNA polymerase can directly add a ribonucleotide to the complementary base pair, the complementary base of the DNA template. This we are saying because at the time of replication that we have already done in another video, the DNA polymerase, it requires the primer. It needs the free hydroxyl at the 3' end, to which the deoxyribonucleotides, they may be attached or binded. The RNA polymerase, it does not require the free 3' hydroxyl group to initiate its activity. So outrightly at this initiation point of initiation, it binds to this DNA double helix, forms the transcription bubble and moves ahead, adding one ribonucleotide that is complementary to the nucleotides present on this template strand, and it moves ahead, resulting in the formation of a RNA molecule. And now this RNA molecule, it is formed in the direction that is 5' to 3' direction, and once this entire stretch of gene or the cistronic unit or the polycistrone, it ends. As soon as it reaches the termination site, the RNA polymerase, it separates from this DNA double helical structure and sets free this RNA. Now I'm saying the gene or the cistron or the polycistron because that depends what is the stretch of the DNA molecule that is supposed to be transcribed by the RNA polymerase that once binds to the promoter region. Now this termination, we are coolly saying that once it reaches the termination site, it sets free this RNA transcript. But actually this termination in case of prokaryotes, it can be it can take place by two pathways. There are two strategies of this termination. One is protein based and the other is RNA based. The intrinsic termination, that is something very natural. It is the RNA based termination of transcription. It does not involve a protein factor, that is the Rho factor. Hence, it is also known as the Rho independent pathway of termination. What happens is that we are saying that once the RNA polymerase, it reaches the termination site. Now, this termination site is marked by the presence of specific nucleotide sequences in the double helical structure in the DNA molecule itself. And this specific structure, this specific nucleotide sequences, they involve two sequences, two stretches of approximately 20 nucleotides which are self-complementary to each other. And this is followed by poly A, that is 7 to 10 nucleotides, the adenine nucleotides. So when the messenger RNA is formed, as soon as the messenger RNA is formed or transcribed on these self-complementary sequences, it base pairs with each other, and this region is guanine cytosine rich region, that is, it is the GC rich region. when it forms when it base pairs with each other, these two complementary sequences, they form this special structure that is known as the stem loop structure or the hairpin loop structure. Presence of this structure at the exit site of RNA polymerase, it loosens the grip of this enzyme RNA polymerase on this DNA double helix. And now these complementary sequences, they are followed by this poly A tail or poly A sequence, and the complementary sequences that will be formed of this RNA molecule, that will be the U, because we know that in RNA molecule instead of thymine uracil is present. So this is the weak bonding region of the RNA DNA hybrid molecule that exists throughout the transcription process within this bubble, a transcription bubble formed by the RNA polymerase. So as soon as it encounters these weak points, this hybrid, it separates from each other. This RNA polymerase, it separates from the DNA double helix, and the regular DNA double helical structure is formed, and this RNA is set free. Now the other mechanism of termination of this transcription in prokaryotes, it depends on the presence of a protein factor, the Rho factor. Now basically this protein, it has got helicase activity, that it opens the helix, and for the purpose it requires ATP. So this Rho factor, this protein that is referred to as the Rho factor, it is an ATP dependent RNA stimulated helicase. Now as this name suggests, this Rho factor, it binds to the RNA molecule that is formed as a result of transcription. Now for the genes in which the termination, it involves this Rho factor, there are certain specific translational stop sequence known as the RUT site on the DNA double helix. RUT stands for Rho utilization point or the Rho utilization site. So when transcription takes place at this RUT site and messenger RNA is formed. Now in case of prokaryotes, one more thing is there, and that is, as soon as this RNA is formed, messenger RNA, the ribosomal machinery, it starts the second process, that is the translation. So when this RUT site is free, that is no ribosome is attached to this RUT site, at that point of time a specific protein known as the Rho factor, it recognizes this RUT site and attaches to it. And once it attaches to it, it moves along the RNA, the newly synthesized RNA, towards the RNA polymerase enzyme. Now one more thing happens, and that is that as soon as this RNA polymerase, it comes across this RUT site on the DNA double helix, its speed, it slows down, and simultaneously this Rho factor, which binds to the RUT site, it moves towards this RNA polymerase, and gradually as it reaches this RNA polymerase, it opens the helical structure between this DNA RNA hybrid, and it sets free the messenger RNA that is being transcribed and sets free this enzyme RNA polymerase. So in this manner, this proteinaceous molecule, the Rho factor, it helps in the termination of the transcription of the particular gene sequence. And the RNA thus formed, the messenger RNA, it is now available in the prokaryotic system for the next process, that is translation. In eukaryotes, this process of transcription, that is formation of RNA on the DNA molecule, is basically the same. This initiation, the elongation and the termination, but it is more complex. And in the upcoming video, we'll be dealing with the initiation of transcription in eukaryotes because it involves many more proteins.